Hello there, and welcome to Midnight Sleep Science. Tonight, we're going to explore something you experience every moment of your life, yet rarely stop to understand: how your brain feels emotions. Right now, as you settle into this quiet space, your brain is processing feelings, perhaps calm, perhaps curiosity, perhaps the gentle weight of the day releasing from your shoulders. These aren't abstract experiences floating somewhere outside you. They're real, measurable events happening inside a 3-pound organ resting behind your eyes. Every emotion you've ever felt, joy, fear, love, sadness, begins as electrical signals traveling between specialized clusters of neurons deep within your brain. These signals trigger chemical messengers called neurotransmitters that flood specific pathways, creating the physical sensations you recognize as feelings. Your racing heart during anxiety, the warmth spreading through your chest when someone you love walks into a room, a tight knot in your stomach before something uncertain. All of these are your brain's way of interpreting the world and preparing your body to respond. Tonight, we'll travel through the hidden architecture of emotion, discovering how ancient brain structures work with modern thinking regions to create the rich emotional landscape of human experience. If you enjoy these gentle journeys, feel free to like and subscribe. But for now, let's begin. Deep beneath the surface of your thinking mind, nestled in the center of your brain, lies a collection of structures so ancient they emerged with the first mammals over 200 million years ago. This is the limbic system, and it's processing your emotions right now, not as a background function, but as your primary interpreter of what matters in the world around you. While you read these words, these primitive structures are continuously scanning for significance, assigning emotional weight to every sensation, every memory, every moment. The limbic system earned its name from the Latin word limbus, meaning border or rim, because these structures form a curved border around the deeper brainstem regions that keep you alive. The French anatomist Paul Broca first noticed this rim of tissue in the 1800s and called it le grande lobe limbique, the great limbic lobe. But it wasn't until the 1950s that neuroscientist Paul MacLean recognized these structures as the brain's emotional command center, the place where feelings originate before conscious thought even begins. MacLean called it the paleomammalian brain because it appeared when early mammals evolved from reptiles, bringing with it an entirely new capacity: the ability to feel. Reptiles respond to their environment with programmed behaviors, flee from larger creatures, approach smaller ones, maintain body temperature. But mammals needed something more sophisticated. They needed to nurse their young, recognize their offspring, form social bonds, distinguish friend from threat in complex social groups. They needed emotions. Your limbic system sits on both sides of the thalamus, tucked beneath the temporal lobes where your conscious thinking happens. If you could slice your brain down the middle and look at the inner surface, you'd see this rim of tissue curving in an arc from front to back, connecting multiple structures that work together as an emotional processing network. The major players in this system include the amygdala, the hippocampus, the hypothalamus, parts of the thalamus, and regions of cortex including the cingulate gyrus. Each structure has specialized functions, but they work as an integrated system, constantly communicating through dense bundles of nerve fibers. When something emotionally significant happens, a sudden loud noise, an unexpected text from someone you love, the smell of a place you haven't visited in years, information flows through these structures in milliseconds, triggering emotional responses before your conscious mind registers what's happening. This speed is crucial. Your limbic system evolved to keep your ancestors alive in dangerous environments where threats appeared suddenly and required immediate action. A rustle in the grass could be wind, or it could be a predator. The mammals who paused to consciously evaluate every stimulus didn't survive long enough to become your ancestors. The ones who felt fear instantly and acted on it, those are the ones whose limbic systems you inherited. The limbic system receives input from all your sensory systems: vision, hearing, touch, smell, taste, but it processes this information differently than your thinking cortex does. Rather than analyzing details or constructing rational interpretations, it evaluates everything for emotional relevance. Is this threatening, rewarding, familiar, novel? Does this matter to my survival, my goals, my relationships? These questions get answered automatically, beneath consciousness, shaping your emotional responses before you even realize you're responding. Consider what happens when you encounter a dog on an empty street. Your visual cortex processes the image, 4 legs, fur, certain size and shape. But simultaneously, your limbic system is already evaluating. Does this animal pose a threat? If you had a frightening experience with a dog years ago, your limbic system remembers. Your amygdala, in particular, stores emotional memories of dangerous situations and instantly recognizes similar patterns. You might feel your heart rate increase and your muscles tense before you consciously think, "I should be careful." That's your limbic system preparing your body to respond, operating on timescales measured in thousandths of a second. But this emotional processing doesn't happen in isolation. The limbic system integrates multiple types of information simultaneously, creating rich emotional experiences that prepare your whole body to respond. At the center of this integration sits the hypothalamus, a structure about the size of an almond that serves as the bridge between your emotional brain and your body's automatic systems. When your amygdala detects a threat, the hypothalamus immediately activates your sympathetic nervous system, the part of your autonomic nervous system responsible for the fight or flight response. Within moments, your hypothalamus sends signals that increase your heart rate, redirect blood flow to your muscles, dilate your pupils, and release stress hormones like cortisol and adrenaline into your bloodstream. These aren't conscious decisions. They're automatic physiological changes orchestrated by this tiny structure that's been performing the same function in mammals for millions of years. Your racing heart during a job interview, the butterflies in your stomach before a first date, the rush of energy when you hear good news. All of these physical sensations of emotion begin with hypothalamic activation. The hypothalamus also regulates basic drives that feel emotional: hunger, thirst, sexual desire, body temperature. These aren't emotions in the traditional sense, but they create powerful motivational states that your brain experiences as feelings. When you feel irritable because you haven't eaten, that's your hypothalamus signaling an imbalance that needs correction. When you feel contentment after a satisfying meal, your hypothalamus has registered that balance has been restored. Sensory information reaches these limbic structures through the thalamus, which sits just above the hypothalamus and acts as a relay station for nearly all sensory input entering your brain. Visual information from your eyes, sounds from your ears, sensations from your skin, they all pass through the thalamus, which then sends this information simultaneously to 2 places: your amygdala for rapid emotional evaluation, and your cortex for slower, more detailed analysis. This dual pathway explains why you can feel afraid before you consciously understand what frightened you. The thalamus to amygdala route is faster than the thalamus to cortex route, giving your limbic system a head start in responding to potential threats. Another crucial limbic structure is the cingulate cortex, a band of tissue that curves along the inner surface of each hemisphere, sitting just above the corpus callosum that connects your brain's 2 sides. The interior portion of the cingulate cortex, the front part, plays a particularly important role in emotion. It receives information from both the amygdala and the orbitofrontal cortex about rewards and punishments, integrating this emotional information with cognitive processes like attention and decision-making. The interior cingulate cortex is where emotion and action connect. When you must choose between competing options, stay home or go out, speak up or stay quiet, take a risk or play it safe, your interior cingulate evaluates the emotional significance of potential outcomes. Research shows that damage to this region impairs people's subjective experience of emotion. They can still recognize emotional situations intellectually, but the feelings themselves become muted, distant. This reveals something profound. The interior cingulate doesn't just process emotional information, it contributes to the actual experience of feeling. The subgenual portion of the cingulate cortex, located below the knee where the structure curves, has direct connections to the hypothalamus and brainstem regions that control autonomic functions. This area helps regulate the physical components of emotion, your heart rate variability, breathing rhythm, stress hormone release. When this region functions normally, it maintains balanced autonomic responses. But disruptions in subgenual cingulate activity are associated with depression, particularly the kind of depression characterized by physical symptoms like fatigue, altered appetite, and disrupted sleep. All these structures, amygdala, hippocampus, hypothalamus, thalamus, cingulate cortex, work as an interconnected network, with dense bundles of nerve fibers allowing constant communication between regions. Emotion emerges from this network activity, not from any single structure in isolation. The system operates on multiple timescales simultaneously: the split-second threat detection of the amygdala, the sustained motivational drives of the hypothalamus, the integration of emotion and memory in the hippocampus, the conscious emotional experience shaped by the cingulate cortex. What makes the limbic system particularly fascinating is how it binds emotion to memory through the partnership between the amygdala and the hippocampus. These two structures sit next to each other in the temporal lobe and work in constant collaboration, ensuring that emotionally significant events are encoded more strongly than neutral ones. When you experience something that triggers a strong emotional response, your amygdala activates and sends signals to your hippocampus, the brain's memory formation center, essentially telling it, "This matters, remember this." Research using direct brain recordings in humans has confirmed this relationship. When people successfully encode emotionally arousing information into memory, high-frequency electrical activity increases in both the hippocampus and amygdala simultaneously. When researchers applied inhibitory electrical stimulation to disrupt this activity, they specifically reversed the memory enhancement normally seen for emotional material. This reveals that the amygdala-hippocampus circuit doesn't just correlate with emotional memory. It causally creates the prioritization that makes emotional experiences more memorable than neutral ones. This is why you remember where you were when you heard devastating news or the vivid details of your wedding day. You can't recall what you ate for lunch 2 weeks ago. The mundane lacks emotional weight, so your hippocampus processes it routinely, often failing to transfer it into long-term storage. But experiences charged with emotion, fear, joy, surprise, love, activate the amygdala, which then modulates hippocampal encoding, ensuring these memories are consolidated and retained. This emotional memory system evolved because survival depends on remembering what matters. A gazelle that fails to remember which watering holes harbor predators won't survive long enough to pass on its genes. A primate that can't recall which group members are trustworthy and which are threats will struggle in complex social hierarchies. Your ancestors' limbic systems learned to prioritize emotional information because emotion signals survival relevance. That ancient prioritization system still operates in you today, determining which moments of your life become permanent memories and which fade into forgotten background noise. The limbic system also connects multiple brain regions through a circuit called the Papez circuit, named after anatomist James Papez, who proposed in 1937 that emotional experience requires communication between cortical and subcortical regions. This circuit forms a loop. The hippocampus sends information through a fiber bundle, called the fornix, to the mammillary bodies, small rounded structures at the base of the hypothalamus. The mammillary bodies then project to the anterior thalamus through the mammillothalamic tract. The thalamus relays this information to the cingulate cortex, which finally sends projections back to the hippocampal formation, completing the circuit. This loop allows emotional information to reverberate through multiple processing levels, creating the rich, multilayered quality of emotional experience. A single event, say seeing an old friend unexpectedly, activates this entire circuit within milliseconds. Your amygdala recognizes the familiar face and attaches positive emotional significance. Your hippocampus retrieves associated memories of past interactions. Your hypothalamus triggers autonomic changes, a smile spreading across your face, warmth in your chest. Your cingulate cortex integrates this emotional information with your current goals and context. The entire limbic network activates as a unified system, creating the complete emotional experience of delighted surprise. These ancient structures, formed when the first mammals walked the earth over 200 million years ago, still govern your emotional life today. They operate continuously, mostly outside awareness, evaluating significance and preparing responses before your conscious mind understands what's happening. Every feeling you experience passes through this ancient system, shaped by structures that evolved to keep your ancestors alive in a dangerous world. And at the very center of this emotional network, constantly vigilant, sits a small almond-shaped structure that deserves closer attention, the amygdala, your brain's sentinel, watching for threats and rewards in every moment. This structure shapes fear, assigns emotional meaning to memories, and triggers responses faster than thought itself. Understanding how it works reveals why emotions feel so immediate, so powerful, and sometimes so difficult to control. Your amygdala sits deep within each temporal lobe of your brain, roughly behind your eyes and just above your ears, shaped like an almond, which is exactly what its name means in Greek. This small structure, barely over a centimeter long, processes some of the most powerful experiences of your life. Fear that makes your heart race, anxiety that tightens your chest, the sudden recognition of danger that happens before you consciously understand what frightened you. The amygdala earned its reputation as your brain's fear center, though this label simplifies what is actually a far more sophisticated system for evaluating emotional significance across all experiences, not just threatening ones. Every moment, your amygdala receives sensory information from the world around you, visual images, sounds, smells, tactile sensations, and performs rapid assessments of emotional meaning. Is this stimulus threatening or safe? Rewarding or punishing? Familiar or novel? Important or ignorable? These evaluations happen in milliseconds, far faster than your conscious awareness can track. The amygdala operates on what neuroscientists call the low road, a fast, automatic pathway that allows emotional responses to begin before your thinking cortex finishes its slower, more detailed analysis. This speed evolved because survival often depends on reacting instantly to threats. When your ancestors encountered a predator on an open plain, the individuals who paused to consciously evaluate whether the approaching shape was dangerous didn't survive. The ones who felt fear immediately and ran, those are your ancestors, and you inherited their amygdala's lightning-fast threat detection system. That same system activates today when you hear a sudden loud noise, when you see someone approach you aggressively, when you stand near the edge of a high place. The amygdala accomplishes this rapid processing through direct connections with sensory regions, particularly the visual thalamus. Visual information from your eyes travels to the thalamus, which then sends signals along two pathways simultaneously. One pathway goes directly to your amygdala. This is the fast, crude route that provides quick but imprecise information. The other pathway travels to your visual cortex for detailed processing before reaching the amygdala. This is the slow, accurate route. The fast pathway allows your amygdala to initiate a response in about 12 milliseconds, while the slow pathway takes several hundred milliseconds. This dual pathway system explains experiences you probably had, jumping at a shape in the dark that turns out to be harmless, or feeling your heart race when someone surprises you even though you quickly recognize them as a friend. Your amygdala reacted to the crude sensory information from the fast pathway, triggering a fear response. Then your cortex processed the detailed information from the slow pathway and sent signals back to your amygdala, essentially saying false alarm. The initial fear response happened before you consciously understood what you were seeing. The amygdala isn't a single unified structure, but rather a complex of 13 distinct nuclei, clusters of neurons with specialized functions. The three main groups are the basolateral complex, the central nucleus, and the cortical nucleus. The basolateral complex receives sensory information and connects extensively with the hippocampus and prefrontal cortex, allowing it to integrate emotional processing with memory formation and higher-order thinking. This is the region that learns which stimuli predict danger or reward through experience. The central nucleus serves as the amygdala's primary output station, sending signals to the hypothalamus and brainstem to trigger the physical manifestations of emotion. When the central nucleus activates, it initiates your fight or flight response. Increased heart rate, rapid breathing, muscle tension, sweating, dilated pupils. These automatic physiological changes happen without conscious decision. Your amygdala simply detects a threat and your body prepares to respond. The amygdala processes more than just fear, though fear remains its most studied function. Research using brain imaging has shown that the amygdala responds to both positive and negative emotional stimuli, with particular sensitivity to faces displaying emotional expressions. When you see someone smile, when you recognize anger in another person's eyes, when you perceive the subtle signs of disgust or sadness in facial expressions, your amygdala is actively processing these emotional signals, often before you consciously register what expression you're seeing. This specialization for emotional faces makes evolutionary sense. For social mammals like humans, reading others' emotions provides crucial information about safety, threat, social status, and relationship dynamics. A face displaying anger signals potential aggression. Your amygdala detects this and prepares your body for possible conflict. A fearful expression in someone you're with suggests danger nearby, even if you haven't seen the threat yourself. Again, your amygdala responds, triggering vigilance. This automatic emotional contagion, where seeing fear in others makes you feel afraid, depends on rapid amygdala processing. The amygdala's role in emotional learning becomes especially clear in fear conditioning, the most extensively studied form of emotional memory formation. In fear conditioning experiments, a neutral stimulus like a tone is paired repeatedly with something unpleasant, such as a mild shock. After just a few pairings, the tone alone begins triggering fear responses, increased heart rate, sweating, freezing behavior in animals, heightened startle responses in humans. This learning happens in the amygdala, specifically in the basolateral complex where sensory information about the tone and the shock converge. When these two types of information arrive simultaneously, the synapses, the connection points between neurons, strengthen through a process called long-term potentiation. This synaptic strengthening creates a memory trace, essentially encoding the association between the tone and danger. Once this association forms, hearing the tone activates the memory trace, triggering activity that flows from the basolateral amygdala to the central nucleus, which then sends signals to the hypothalamus and brainstem to produce the physical fear response. Research using precise lesions in animals has confirmed the amygdala's essential role in this process. Damage to the lateral nucleus prevents animals from forming new fear associations. They can still feel pain from the shock and hear the tone, but they cannot learn that the tone predicts danger. Damage to the central nucleus produces a different effect. Animals can form the association but cannot express the appropriate fear responses. This demonstrates that different parts of the amygdala handle learning versus expression of conditioned fear. In humans, studies of people with amygdala damage reveal similar patterns. These individuals show impaired emotional memory. They remember events, but the memories lack the emotional intensity that normally accompanies significant experiences. When shown disturbing images that would normally create lasting memories in people with intact amygdalae, patients with amygdala damage remember these images no better than neutral ones. This suggests the amygdala doesn't store the memories themselves, but rather enhances memory formation in other brain regions like the hippocampus by tagging emotionally significant information as important. This enhancement happens through the amygdala's extensive connections to memory systems. During emotionally arousing experiences, the amygdala releases modulatory signals that strengthen memory consolidation processes in hippocampus and cortex. Stress hormones released during emotional events activate receptors in the amygdala, which in turn influences memory storage elsewhere. This is why you remember emotionally charged moments with such clarity. Your amygdala ensured those memories were prioritized for storage. Studies of patients with amygdala damage also reveal diminished emotional arousal, particularly for negative stimuli. When shown disturbing images, these patients accurately recognize the images as negative and can categorize them correctly, showing their cognitive understanding remains intact. But they don't show the typical physiological arousal responses, the increased heart rate, skin conductance changes, and subjective feelings of distress that people with functioning amygdalae experience. Their emotional knowledge remains, but the automatic emotional reaction, the arousal that makes emotions feel urgent and important, diminishes. This separation between emotional knowledge and emotional experience reveals something profound about the amygdala's function. The amygdala doesn't just help you recognize emotions intellectually. It generates the arousal component that makes emotions feel real, that creates the visceral experience of fear, excitement, or intense joy. Without this arousal, emotional life becomes muted, distant, more cognitive than felt. The amygdala's speed can sometimes work against you in modern life, a phenomenon psychologist Daniel Goleman famously termed amygdala hijack. This describes moments when your amygdala responds so quickly and powerfully to a perceived threat that it essentially overrides your rational thinking before your prefrontal cortex can evaluate whether the threat is genuine. You might snap at someone in anger, burst into tears, or freeze completely, reactions you later regret when your thinking brain catches up and realizes the situation didn't warrant such intensity. These hijacks happen because the amygdala's threat detection system evolved in an environment where dangers were often immediate and physical: predators, aggressive competitors, environmental hazards. Your amygdala still operates as if you live in that ancestral world, treating social threats like criticism, rejection, or embarrassment as if they were physical dangers requiring immediate defensive action. A harsh comment from a colleague can trigger the same physiological cascade: racing heart, tense muscles, stress hormone release, that would have prepared your ancestors to flee from a predator. Individual differences in amygdala reactivity help explain why people vary so dramatically in their emotional responses to the same situations. Research shows that individuals with high trait anxiety, a tendency to experience anxiety across many situations, display exaggerated automatic reactivity in their amygdala when processing fearful faces, even when those faces are shown so briefly they don't reach conscious awareness. This heightened sensitivity means their threat detection system activates more easily and intensely, creating a vulnerability to anxiety disorders and stress-related problems. Brain imaging studies have found that anxious individuals show increased amygdala responses to threatening stimuli that wouldn't produce such strong reactions in people with lower anxiety. This suggests anxiety isn't just about thinking anxious thoughts. It reflects a fundamental difference in how the amygdala evaluates potential threats. For someone with a highly reactive amygdala, the world genuinely feels more threatening because their emotional sentinel is set to maximum sensitivity, detecting danger where others perceive neutral or mildly concerning situations. The amygdala also processes positive emotions, though this role receives less attention than its fear functions. Recent research has discovered that the central nucleus of the amygdala, long associated primarily with fear responses, actually contains abundant circuits that respond to rewarding events. When these reward circuits activate in mice, the animals seek out those stimuli again, demonstrating that the amygdala drives approach behavior toward pleasant experiences, not just avoidance of threatening ones. Studies using functional brain imaging in humans have confirmed that both the left and right amygdala activate in response to intensely positive emotional stimuli: beautiful images, joyful scenes, pleasurable experiences. The key factor isn't whether the emotion is positive or negative, but rather its intensity. The amygdala functions as an intensity detector, responding more strongly as emotional arousal increases, regardless of whether that arousal comes from joy or fear, pleasure or pain. This makes evolutionary sense. Your brain needs to pay attention to and remember intense experiences of any kind, because both extremely good and extremely bad events carry important information for survival. When the amygdala detects emotional significance, whether threatening or rewarding, it doesn't act alone. It triggers cascades of chemical messengers, neurotransmitters, that flow through specific pathways, creating the physical sensations and subjective feelings you experience as emotions. These chemicals paint your emotional world with different colors and textures, determining whether you feel energized joy or crushing sadness, sharp fear or nauseating disgust. Understanding these neurotransmitters reveals the biochemical foundation of feeling, the molecular mechanisms that translate neural activity into conscious emotional experience. Between every thought you think, every movement you make, every emotion you feel, lies a microscopic gap called a synapse. This space, measuring only about 20 to 40 nanometers across, roughly 1,000th the width of a human hair, separates one neuron from the next. Your brain contains approximately 100 billion neurons, and each neuron may connect with thousands of others, creating trillions of these tiny gaps. Information cannot cross these gaps as electrical signals alone. Instead, your neurons rely on chemical messengers called neurotransmitters to carry information across synapses, translating electrical activity in one neuron into chemical signals that the next neuron can receive. When an electrical signal reaches the end of a neuron's axon, the long projection that sends messages to other cells, it triggers a cascade of events at the synapse. Calcium ions flood into the neuron's terminal, causing tiny spherical packages called vesicles to fuse with the cell membrane. These vesicles contain thousands of neurotransmitter molecules that have been synthesized and stored, waiting for this moment. When the vesicles merge with the membrane, they release their contents into the synaptic gap, a process called exocytosis. The neurotransmitters then diffuse across the synapse, a journey measured in millionths of a second, until they encounter receptor proteins on the surface of the receiving neuron. These receptors are exquisitely specific, shaped to accept only certain neurotransmitter molecules, like locks that only particular keys can open. When a neurotransmitter molecule binds to its matching receptor, it changes the receptor shape, opening ion channels that allow charged particles to flow into or out of the receiving neuron. This changes the electrical state of that neuron, making it either more or less likely to fire its own signal. After transmitting their message, neurotransmitters don't remain in the synapse indefinitely. Some are broken down by enzymes floating in the synaptic space. Others are recaptured by the sending neuron through transporter proteins that pull them back inside, where they can be repackaged into vesicles and used again, a recycling process called reuptake. This efficient system allows your brain to maintain precise control over neurotransmitter levels, fine-tuning the strength and duration of chemical signals. Your brain produces dozens of different neurotransmitters, each with specialized functions. Some are excitatory, making the receiving neuron more likely to fire. Others are inhibitory, making it less likely to fire. Some neurotransmitters work quickly and locally, affecting only nearby neurons. Others act more broadly as neuromodulators, released from multiple sites throughout the brain to influence the activity of millions of neurons simultaneously. 3 neurotransmitters in particular, dopamine, serotonin, and norepinephrine, play central roles in your emotional life. These chemicals belong to a family called monoamine neurotransmitters, named for their molecular structure containing a single amine group. Researchers have proposed what they call the 3 primary color model of basic emotions, suggesting these 3 monoamines work together like primary colors mixing to create the full spectrum of human feeling. According to this model, dopamine mediates experiences of reward and joy. Serotonin relates to punishment, disgust, and sadness. Norepinephrine drives stress responses, fear, and anger. Just as red, blue, and yellow can combine in countless proportions to create every color imaginable, these 3 neurotransmitters blend in varying ratios to produce the complex emotional states you experience throughout each day. Recent groundbreaking research has confirmed that these neurotransmitters are released specifically when processing emotional content, even in language. When you read emotionally charged words or hear someone speak with emotional intensity, your brain releases dopamine and serotonin in specific regions that process emotional meaning. This reveals that these chemical messengers don't just respond to external events. They help your brain interpret the significance of symbols and communication, functions uniquely important to human survival. The balance of these neurotransmitters profoundly influences your emotional baseline and your vulnerability to mood disorders. Low serotonin levels have been consistently associated with depression and anxiety. Disrupted dopamine function relates to anhedonia, the inability to feel pleasure, and apathy. Dysregulated norepinephrine contributes to anxiety disorders and depression. Most psychiatric medications work by altering the levels or activity of these 3 neurotransmitters, either by blocking their reuptake, preventing their breakdown, or mimicking their effects at receptors. Dopamine is perhaps the most widely recognized of these 3 messengers, often called the feel-good neurotransmitter. It's produced primarily in the ventral tegmental area deep in your midbrain and travels along pathways called the mesolimbic and mesocortical systems to regions including the nucleus accumbens, amygdala, hippocampus, and prefrontal cortex. When you encounter something rewarding, delicious food, a compliment from someone you respect, progress toward a meaningful goal, dopamine neurons in the ventral tegmental area fire rapidly, releasing dopamine into the nucleus accumbens. This release creates the subjective experience of pleasure and simultaneously signals your brain, "This is good, remember this, do it again." This reward system evolved to motivate behaviors essential for survival. Your ancestors who experienced pleasure from eating high-calorie foods, forming social bonds, and exploring new environments were more likely to survive and reproduce. The dopamine system reinforced these beneficial behaviors by making them feel good. That same system still operates in you today, creating motivation and pleasure when you achieve goals, connect with others, or discover something novel. But dopamine doesn't just respond to receiving rewards. It also activates in anticipation. When you expect something pleasurable, when you work toward a goal, dopamine release creates a feeling of eagerness and motivation. This anticipatory dopamine is why the journey toward a goal often feels more emotionally intense than achieving the goal itself. Your brain releases dopamine throughout the pursuit, driving you forward with feelings of hope and excitement. Serotonin, by contrast, creates very different emotional tones. This neurotransmitter is synthesized mainly in the raphe nuclei of the brainstem and projects throughout the brain, but more than 90% of your body's serotonin actually exists in your gastrointestinal tract. In the brain, serotonin acts as a master regulator of mood, affecting emotional processing, attention, and behavioral inhibition. When serotonin levels are balanced, people report feeling emotionally stable, calm, and focused. Low serotonin has been consistently linked to depression, anxiety, impulsivity, and aggression. Recent research suggests serotonin may be particularly involved in processing punishment, disgust, and aversive experiences. When you encounter something disgusting or disappointing, serotonin activity changes in ways that create withdrawal responses and behavioral inhibition. Studies have shown that depleting tryptophan, the amino acid precursor your brain uses to make serotonin, enhances emotional reactions to social conflict and amplifies feelings of guilt in highly empathic individuals. This reveals that serotonin helps regulate how intensely you respond to negative social and emotional situations. Norepinephrine, the third primary emotional neurotransmitter, mediates arousal, alertness, and the stress response. It's produced in the locus coeruleus, a tiny structure in the brainstem that projects to nearly every region of your brain. Norepinephrine release increases during any situation requiring vigilance or action. When you face a challenge, encounter a potential threat, or need to focus intensely on something important. This neurotransmitter drives the fight or flight response. When your amygdala detects danger, it triggers the locus coeruleus to flood your brain with norepinephrine while simultaneously triggering your adrenal glands to release epinephrine, adrenaline, into your bloodstream. Together, these catecholamines create the characteristic physical sensations of acute stress: racing heart, rapid breathing, sharpened focus, heightened alertness. Your pupils dilate, blood flow redirects to your muscles, glucose mobilizes from storage to fuel immediate action. Your entire body prepares to respond decisively to whatever threat triggered this cascade. These three neurotransmitters don't work in isolation, but blend together like primary colors mixing to create the full emotional spectrum. Joy without arousal feels different from excited joy. The first involves primarily dopamine, the second combines dopamine with norepinephrine. Sadness tinged with anxiety involves both serotonin and norepinephrine, creating a distinctly different feeling than pure melancholy. Every complex emotional state you experience emerges from varying proportions of these three chemical systems interacting with each other, and with your memories, thoughts, and current circumstances. Think of an artist's palette where three primary colors, red, blue, and yellow, can combine in infinite proportions to create every shade imaginable. Your emotional brain operates on a remarkably similar principle, using three primary neurotransmitter systems to generate the full spectrum of human feeling. Dopamine, serotonin, and norepinephrine blend together in varying concentrations, creating emotions as distinct as pure joy, bittersweet nostalgia, anxious excitement, or crushing despair. This three primary color model of basic emotions suggests that understanding how these chemical systems combine reveals why emotions feel so varied, so nuanced, so impossibly complex. Dopamine forms the first primary color in this model, representing the dimension of reward, pleasure, and approach motivation. When dopamine dominates your neurochemical landscape with relatively lower contributions from serotonin and norepinephrine, you experience something close to pure joy, the delight of receiving good news, the satisfaction of completing a challenging project, the warmth of being with people you love. This dopamine-driven joy feels light, energizing, approach-oriented. Your body feels drawn toward the source of pleasure, wanting more of whatever triggered this rewarding state. But dopamine rarely operates alone. Add norepinephrine to high dopamine levels, and the quality of joy transforms into excitement, that buzzing, energized feeling of anticipation before something wonderful. Your heart races, your attention sharpens, you feel almost electrically alert. This is the emotion you feel before a first date, before stepping on stage to perform, before opening a gift you've been waiting for. The norepinephrine adds arousal and vigilance to dopamine's pleasure signal, creating a more intense, more physiologically activated form of positive emotion. Serotonin represents the second primary color, associated with punishment, aversion, and behavioral inhibition. When serotonin activity is balanced and moderate, it creates feelings of contentment, calm, and emotional stability, not the excited joy of high dopamine, but rather a quiet satisfaction with things as they are. But when experiences trigger strong aversive serotonin responses with low dopamine, you feel disgust, disappointment, or sadness. These emotions feel heavy, withdrawal-oriented. Your body wants to move away from whatever triggered this state, to avoid or escape rather than approach. Research has shown that depleting serotonin amplifies responses to social punishment and increases feelings of guilt and shame in people with high empathy. This reveals serotonin's role in processing social rejection and moral violations, experiences that feel distinctly different from physical threats but nonetheless trigger powerful emotional responses. The sadness of disappointing someone you respect, the disgust at witnessing injustice, the shame of failing to meet your own standards, these complex social emotions depend on serotonin's aversive signaling. Norepinephrine forms the third primary color, mediating arousal, stress, and fear responses. When norepinephrine surges with relatively lower dopamine and serotonin, you experience something close to pure fear or anxiety, that acute, vigilant, threat-focused state where your body prepares for immediate defensive action. Your amygdala has detected danger and triggered your locus coeruleus to flood your brain with norepinephrine, while your adrenal glands release epinephrine into your bloodstream. Together, these catecholamines create the unmistakable physical signature of fear: racing heart, rapid, shallow breathing, sweating palms, tense muscles ready to fight or flee. But mixed norepinephrine with serotonin's aversive signals and fear transforms into anxiety tinged with sadness, that heavy, worried, defeated feeling when a threat feels both imminent and insurmountable. This combination creates the emotional tone of depression with agitation, where you feel simultaneously threatened and hopeless, aroused and exhausted. Add some dopamine to high norepinephrine, and you get something entirely different: the aggressive energy of anger, where threat detection combines with approach motivation to create fighting rather than fleeing. These three neurotransmitters don't just create isolated emotions, they combine simultaneously to produce the complex, layered feelings that characterize real human experience. Research measuring actual neurotransmitter release in human brains during emotional processing has confirmed that emotionally charged words trigger simultaneous dopamine and serotonin release in specific brain regions, with a pattern varying based on whether the words are positive or negative. This reveals that even processing language with emotional meaning activates these chemical systems in coordinated ways. The emotional dimension psychologists describe, valence, and arousal, map directly onto these neurotransmitter systems. Valence refers to whether an emotion feels pleasant or unpleasant, ranging from intensely positive to intensely negative. This dimension corresponds primarily to the balance between dopamine's rewarding signals and serotonin's aversive signals. High dopamine with low serotonin creates positive valence, joy, excitement, satisfaction. Low dopamine with high serotonin creates negative valence, sadness, disgust, disappointment. Arousal represents the second dimension, how calm or energized you feel, ranging from deeply relaxed to intensely activated. Norepinephrine primarily determines this dimension. Low norepinephrine creates calm states regardless of valence, peaceful contentment when combined with positive valence, quiet sadness when combined with negative valence. High norepinephrine creates aroused states, excited joy when combined with positive valence, anxious fear when combined with negative valence. Brain imaging studies have confirmed that different neural circuits process these two dimensions. Activity in the amygdala correlates with valence, particularly negative valence, while activity in the thalamus and frontomedial regions varies with arousal. The anterior insula shows increased activity with negative valence, while somatosensory regions reflect the peripheral physiological changes that accompany high arousal. These findings suggest your brain represents emotions along these two fundamental axes, with different neurochemical and neural systems handling each dimension. The real richness of emotional life emerges when you consider that these three systems can activate in countless different proportions. When all three neurotransmitters are at moderate levels, you experience a balanced, neutral state, alert but not stressed, content but not euphoric, neither drawn toward nor repelled by your current situation. Shift the balance slightly, and entirely different feelings emerge. High dopamine and serotonin with low norepinephrine might create a contemplative satisfaction, pleased with life but calm, engaged but not aroused. Low dopamine and serotonin with high norepinephrine creates the jittery, unpleasant feeling of anxiety without clear cause, aroused and vigilant but lacking both reward and punishment signals to explain why. Recent research has even identified the neural signatures of mixed emotions, those complex states where you feel opposing feelings simultaneously. Studies using brain imaging while people watch bittersweet films show that mixed emotions activate unique patterns in cortical regions like the anterior cingulate cortex and ventromedial prefrontal cortex, but not in deeper structures like the amygdala or insula. This suggests that simpler emotional states activate lower brain regions that process positive and negative as mutually exclusive, while more sophisticated cortical areas can integrate contradictory emotional information into genuinely mixed feelings. The feeling of nostalgia perfectly illustrates this neurochemical mixing. You remember something wonderful from your past. High dopamine signals reward as you recall those positive experiences. But simultaneously, you recognize that moment is gone forever, triggering serotonin's aversive processing of loss. The result feels distinctly different from pure joy or pure sadness. It's genuinely mixed, a sweet ache that pulls you toward the memory while simultaneously reminding you it exists only in the past. Your anterior cingulate cortex and ventromedial prefrontal cortex hold both feelings together, creating emotional complexity that simpler brain regions cannot achieve. This three-color model explains why emotions feel so varied despite involving just three primary neurotransmitter systems. Like mixing red, blue, and yellow in different ratios creates orange, purple, green, and every intermediate shade, combining dopamine, serotonin, and norepinephrine in varying proportions creates the entire emotional spectrum humans experience. Understanding this chemical foundation reveals why emotions aren't just abstract feelings floating in consciousness. They're physical, measurable events unfolding in your brain, shaped by molecular signals evolved over millions of years to guide behavior and promote survival. Understanding this neurochemical foundation helps explain why psychiatric medications work the way they do. Depression isn't simply being sad. It reflects specific disruptions in these three neurotransmitter systems. Low serotonin contributes to negative mood, sleep disturbances, and loss of interest in activities. Depleted dopamine creates anhedonia, the inability to feel pleasure from things that should be rewarding. Dysregulated norepinephrine produces fatigue, difficulty concentrating, and disrupted stress responses. Most antidepressant medications work by increasing the availability of these neurotransmitters at synapses. Selective serotonin reuptake inhibitors, the most commonly prescribed antidepressants, block the transporters that would normally recapture serotonin from the synaptic gap, allowing serotonin to remain active longer and compensate for low production. Other medications affect multiple neurotransmitter systems simultaneously, serotonin and norepinephrine, or dopamine and norepinephrine, matching the treatment to the patient's specific symptom profile. Research has even begun mapping specific symptoms to specific neurotransmitters, allowing more targeted treatment. Symptoms like sadness, guilt, and suicidal thoughts correlate particularly with serotonin dysfunction. Fatigue, concentration problems, and psychomotor retardation, slowed movements and thoughts, relate more to norepinephrine. Loss of pleasure, lack of motivation, and emotional numbness connect to dopamine depletion. This symptom-specific approach suggests that understanding which neurotransmitter systems are disrupted could guide selection of medications most likely to help individual patients. But neurotransmitters alone don't create the conscious experience of emotion. These chemical messengers trigger activity in limbic structures like the amygdala and hypothalamus, creating rapid, automatic responses. Yet the subjective feeling, the awareness of being afraid, the recognition that you feel joyful, the ability to name and describe your emotional state, requires something more. These raw emotional signals must reach your cortex, where they transform from automatic reactions into conscious experiences you can think about, talk about, and intentionally regulate. The amygdala can process emotional stimuli even when they don't reach conscious awareness. Fearful faces flash too briefly to perceive consciously still activate amygdala responses. But awareness of emotion requires interaction between subcortical structures and cortical networks that construct conscious experience. Recent theories propose that emotional consciousness emerges when cortical circuits assemble higher-order representations of the lower-order signals coming from the amygdala, hypothalamus, and brainstem neurotransmitter systems. This transformation from automatic emotional reaction to conscious emotional feeling happens in specific cortical regions that integrate subcortical signals with memories, context, language, and self-awareness. The anterior cingulate cortex, the insula, and portions of the prefrontal cortex receive inputs from limbic structures and weave them into the rich, conscious emotional experiences that define human feeling. Understanding how this conscious awareness emerges reveals the fundamental difference between having an emotional response and feeling an emotion, a distinction that helps explain how humans can sometimes regulate feelings that other animals simply react to automatically. Your amygdala can detect a threat and trigger a fear response before you consciously realize anything frightening has happened. Your hypothalamus can activate stress hormones and accelerate your heart rate without any deliberate decision on your part. Neurotransmitters can flood specific pathways, creating physiological states of arousal or calm entirely outside your awareness. These automatic processes happen continuously, generating emotional reactions every moment. Yet you don't just react emotionally, you feel emotions consciously. You can identify that you're anxious, name the feeling as fear, describe the sensation of joy spreading through your chest. This conscious awareness of emotion requires something beyond the limbic system's automatic responses. It requires your cortex. The cortex, the wrinkled outer layer of your brain, represents the most recent evolutionary development in your nervous system. While limbic structures like the amygdala emerged with early mammals over 200 million years ago, the massive expansion of cortical tissue that characterizes human brains happened far more recently. This expansion brought unprecedented cognitive abilities, abstract reasoning, language, long-term planning, self-awareness. It also fundamentally transformed emotional experience. Rather than simply reacting to emotional triggers, cortical processing allows you to be aware that you're having an emotional reaction, to reflect on why you feel the way you do, to consider whether your emotional response matches the situation, and to choose whether to act on your feelings. The transformation from automatic emotional reaction to conscious emotional feeling happens as signals from your limbic system propagate upward through increasingly sophisticated levels of cortical processing. The amygdala detects emotional significance, and triggers rapid responses through its connections to the hypothalamus and brainstem. But simultaneously, the amygdala sends information to cortical regions that integrate these emotional signals with sensory information, memories, contextual understanding, and self-related processing. This cortical integration creates the rich, conscious experience of emotion that feels uniquely human. Three cortical regions play particularly crucial roles in emotional consciousness: the anterior cingulate cortex, the insula, and portions of the prefrontal cortex. These areas don't simply receive emotional information passively, they actively construct your subjective experience of feeling, weaving together bodily sensations, contextual information, memories, and conceptual knowledge to create the conscious awareness of being in an emotional state. The anterior cingulate cortex, which you encountered earlier as part of the limbic system, forms a crucial bridge between subcortical emotional processing and conscious awareness. This band of tissue curves along the inner surface of each hemisphere, positioned perfectly to integrate signals from the amygdala below with cognitive processing in the prefrontal cortex above. The anterior portion, the front part, receives dense inputs from both the amygdala and the ventromedial prefrontal cortex. When these inputs converge, the anterior cingulate cortex evaluates the emotional significance of events and helps translate automatic reactions into conscious feelings. Research using brain imaging has shown that the anterior cingulate cortex activates strongly during emotional experiences, particularly when those experiences involve some degree of conflict or difficulty. When you must choose between emotionally charged options, when you experience pain, when you detect errors or unexpected outcomes, your anterior cingulate cortex shows increased activity. This region appears to monitor for situations that require emotional attention and adjustment, signaling when automatic responses may not suffice and conscious emotional processing needs to engage. People with damage to the anterior cingulate cortex show a peculiar pattern. They can recognize emotional situations intellectually and describe what emotions would be appropriate, but their subjective experience of emotion becomes muted and distant. They understand that a situation should feel frightening or joyful, but the actual feeling lacks its normal intensity. This reveals that the anterior cingulate cortex doesn't just process emotional information, it contributes to the felt quality of emotion itself, the subjective intensity that makes feelings viscerally real rather than merely conceptual. The insula, another critical region for emotional consciousness, sits deep within the lateral sulcus, the fold separating your temporal lobe from your frontal and parietal lobes. The insula receives extensive input from your body, particularly signals about your internal physiological state: heart rate, breathing, digestive processes, muscle tension, temperature. This region essentially creates a map of your body's internal condition moment by moment. What makes the insula crucial for emotional consciousness is how it integrates these bodily signals with emotional information from the amygdala. When your amygdala detects something emotionally significant, your body's state changes, your heart rate increases or decreases, your breathing pattern shifts, your muscles tense or relax, stress hormones enter your bloodstream. The insula receives detailed information about all these bodily changes. It then integrates this interoceptive information, awareness of internal bodily states, with the emotional significant signals from the amygdala, creating the felt bodily experience of emotion. Studies of people with insula damage show impaired emotional awareness, particularly for their own feelings. These individuals struggle to identify what emotion they're experiencing, often describing general arousal or discomfort without being able to name specific feelings. They can see emotional expressions in others and know intellectually what emotions mean, but accessing their own emotional states becomes difficult. This suggests the insula plays an essential role in bringing emotional experience into consciousness by mapping emotions onto bodily sensations that you can introspect and recognize. The prefrontal cortex, the frontmost portion of your brain, sitting right behind your forehead, represents the highest level of emotional processing. Different regions within the prefrontal cortex serve distinct functions in emotional consciousness and regulation. The ventromedial prefrontal cortex, located at the bottom front of the brain, helps assign emotional values to different options when making decisions. The dorsolateral prefrontal cortex, on the outer sides, implements cognitive strategies for regulating emotions. The anterior prefrontal cortex, at the very front, engages in abstract emotional reasoning and reappraisal. Recent research has revealed that emotion regulation, the ability to consciously modulate your emotional responses, involves particular areas of the anterior prefrontal cortex and other higher cortical hierarchies. When people are asked to recontextualize negative images to make them less aversive, these high-level prefrontal regions activate strongly. The more people activate these emotion regulation selective brain regions, the more resilient they are to negative experiences. These findings link these prefrontal areas to better mental health and the ability to resist temptations and avoid addiction. Interestingly, while these prefrontal regions regulate emotional responses, the amygdala responds to aversive experiences the same way, whether people are actively using thoughts to self-regulate their emotions or not. This reveals that it's really the cortex that generates people's emotional responses by changing how they see and attach meaning to events in their environments. The prefrontal cortex doesn't suppress the amygdala directly. Instead, it reframes situations, altering their emotional meaning before the amygdala has fully processed them. The interaction between these cortical regions and subcortical structures creates what neuroscientists call a higher-order theory of emotional consciousness. According to this framework, emotional consciousness emerges when cortical circuits construct higher-order representations of the lower-order emotional signals coming from structures like the amygdala, hypothalamus, and brainstem neurotransmitter systems. The amygdala might detect threat and trigger a fear response, but conscious awareness that you feel afraid requires cortical regions to represent that amygdala activity in a form accessible to conscious thought, language, and deliberate reasoning. This explains why emotions can influence behavior even when they don't reach consciousness, yet conscious feelings provide something uniquely powerful: the ability to think about emotions, talk about them, understand them, and potentially change them. The cortical transformation of automatic emotional reactions into conscious feelings creates a new kind of emotional flexibility unavailable to species with less developed cortical systems. Understanding this cortical processing reveals how humans gained a remarkable capacity to regulate emotions that once regulated them, a capacity that distinguishes human emotional life from the more rigid emotional responses of other animals. The capacity to regulate emotion, to consciously modulate feelings that might otherwise overwhelm you, represents one of the most remarkable achievements of the human brain. When your amygdala detects a threat and triggers a surge of fear, when disappointment threatens to spiral into despair, when anger rises in response to perceived injustice, your prefrontal cortex can step in and alter the trajectory of these emotional responses. This isn't suppression, pushing feelings down while they continue churning beneath the surface. True emotion regulation involves changing how you interpret situations, shifting attention away from emotional triggers, or reframing events to alter their emotional meaning before the full emotional response unfolds. Your prefrontal cortex, that massive expanse of cortical tissue sitting right behind your forehead, acts as your brain's emotional conductor, coordinating and modulating the raw emotional signals generated by limbic structures. This region doesn't develop fully until your mid-20s, which helps explain why children and adolescents struggle with emotional control despite having perfectly functional amygdalae and other emotional processing structures. The regulatory capacity of the prefrontal cortex represents a late-arriving evolutionary and developmental achievement, layering sophisticated control over ancient emotional systems that operated for millions of years without such oversight. The prefrontal cortex regulates emotion through its dense connections with the amygdala and other limbic structures. When emotional hijacking occurs, when your amygdala responds so quickly and powerfully that it threatens to override rational thought, the prefrontal cortex can inhibit amygdala activity, essentially applying the brakes to emotional responses that have begun but haven't yet fully developed. Brain imaging studies show that when people successfully regulate negative emotions, activity in prefrontal regions increases while activity in the amygdala decreases. This inverse relationship reveals the regulatory dynamic. The more your prefrontal cortex engages with emotional situations, the less your amygdala drives automatic reactions. But this regulation doesn't happen through simple suppression. Research comparing different emotion regulation strategies has revealed important distinctions. Suppression, trying to hide or inhibit emotional expressions without changing the underlying feeling, activates the prefrontal cortex late in the emotional response after feelings have already developed. This strategy requires continuous effort to maintain the suppression and often leads to increased physiological arousal even as outward expressions diminish. People who habitually use suppression report less life satisfaction and more negative emotions overall. Reappraisal, by contrast, represents a far more effective regulatory strategy. This approach involves changing how you think about a situation before the full emotional response develops, reinterpreting events to alter their emotional significance. When you reappraise, your prefrontal cortex activates early in the emotional response before the amygdala has fully processed the emotional stimulus. By changing the meaning of the situation, viewing a criticism as helpful feedback rather than a personal attack, seeing a setback as a learning opportunity rather than a failure, you alter the emotional stimulus itself. The amygdala then responds to this reinterpreted meaning rather than the initial interpretation, naturally generating a different emotional response. Recent research has mapped specific prefrontal regions involved in emotion regulation with remarkable precision. The ventromedial prefrontal cortex, located at the bottom front of your brain, plays a crucial role in evaluating emotional stimuli and encoding their significance. When you encounter emotionally charged situations, this region integrates information from the amygdala with contextual details and memories, creating a comprehensive evaluation of the situation's emotional meaning. Under stress, the ventromedial prefrontal cortex shows strong activation in response to highly emotional images, and individual differences in this activation pattern correlate with how people subjectively respond to stress. The dorsolateral prefrontal cortex, situated on the outer sides of your frontal lobes, implements cognitive control strategies during emotion regulation. This region activates when you consciously redirect attention away from emotional stimuli, when you apply cognitive reappraisal strategies to reinterpret situations, or when you deliberately inhibit emotional impulses. Studies have shown that people who show greater activation in dorsolateral prefrontal regions during emotion regulation tasks report better mental health, greater resilience to negative experiences, and improved ability to resist temptations and avoid addictive behaviors. The anterior prefrontal cortex, the very frontmost portion of your brain, engages in more abstract forms of emotional reasoning and regulation. This region helps you step back from immediate emotional reactions to consider broader contexts, long-term goals, and alternative perspectives. When you pause during an argument to consider whether responding with anger will damage an important relationship, when you acknowledge feeling anxious but choose to proceed anyway because the goal matters more than the discomfort, when you recognize that your emotional reaction might be disproportionate to the actual situation, these sophisticated regulatory processes depend on anterior prefrontal involvement. Different prefrontal subregions preferentially process different types of emotional content. Some areas specialize in assigning value to external sensory information, others to memories and imagined future events, still others to internal bodily signals or ongoing emotional states. This content specialization means that emotion regulation isn't a single unified process but rather a collection of related processes, each implemented by different prefrontal circuits depending on what aspect of emotion needs regulating. Functional connectivity between the prefrontal cortex and subcortical regions reveals the neural pathways through which regulation occurs. When people successfully regulate emotions during stress, connectivity between the ventromedial prefrontal cortex and the midbrain, particularly regions like the locus coeruleus that release norepinephrine, shows characteristic patterns. Stronger negative connectivity, where increased prefrontal activity corresponds to decreased midbrain activity, predicts better stress regulation. This suggests the prefrontal cortex can modulate not just amygdala responses, but also the neurotransmitter systems that create the physiological foundation of emotional arousal. Mindfulness meditation has been shown to strengthen connections between the prefrontal cortex and amygdala, potentially improving emotional regulation through concrete neurobiological changes. Regular meditation practice doesn't just teach cognitive strategies. It appears to physically alter the functional connectivity between regulatory and emotional processing regions. This finding has significant implications for mental health treatments, suggesting the practices emphasizing present moment awareness and non-judgmental observation of emotions might enhance emotion regulation by strengthening the neural circuits that implement it. The development of emotional regulation capabilities follows a protracted timeline extending into the mid-20s. While the amygdala reaches functional maturity relatively early in development, prefrontal regions continue developing throughout childhood, adolescence, and early adulthood. This developmental asynchrony, mature emotional processing regions operating alongside still maturing regulatory regions, helps explain the emotional intensity and regulatory difficulties that characterize adolescence. Teenagers aren't simply choosing to be emotionally volatile. Their brains genuinely lack the fully developed regulatory systems that allow adults to modulate emotional responses more effectively. Individual differences in emotion regulation capacity show clear neural correlates. People who report greater difficulty managing emotions show reduced activation in prefrontal regulatory regions and altered connectivity patterns between prefrontal and limbic structures. Emotional dysregulation, persistent difficulty modulating emotional intensity or duration, characterizes many psychiatric conditions including depression, anxiety disorders, bipolar disorder, and borderline personality disorder. Understanding the neural basis of these regulatory difficulties has opened new avenues for treatment, including approaches that specifically target strengthening prefrontal regulatory circuits. The balance between emotional arousal and regulation represents a delicate dynamic that shifts moment by moment. Sometimes rapid amygdala responses serve you well, alerting you to genuine threats or opportunities that require immediate action. Other times, automatic emotional reactions prove maladaptive, generating fear of situations that aren't truly dangerous or anger disproportionate to minor slights. The prefrontal cortex's regulatory capacity allows flexibility. You can choose when to trust rapid emotional responses and when to engage slower, more deliberate evaluation. This regulatory flexibility transforms emotional experience from something that simply happens to you into something you can actively shape. The same neural architecture that generates automatic emotional responses, amygdala threat detection, hypothalamic activation of stress responses, neurotransmitter floods that create physiological arousal, remains active and functional. But the addition of prefrontal regulatory capacity creates a new layer of control, allowing you to intervene in emotional processes that would otherwise run their automatic course. Interestingly, research has shown that the amygdala responds to aversive experiences the same way, whether people actively regulate their emotions or not. The amygdala detects threats and assigns emotional significance regardless of what regulatory strategies you employ. This reveals something profound. Successful emotion regulation doesn't primarily work by suppressing the amygdala's activity directly. Instead, the prefrontal cortex changes how you interpret and understand situations, altering their emotional meaning before the amygdala has fully processed them. By the time emotional information reaches your amygdala, it's already been reframed, recontextualized, or reinterpreted. This explains why cognitive reappraisal proves more effective than suppression. Reappraisal changes the emotional stimulus itself at the level of meaning. When you reinterpret a stressful presentation as an exciting challenge rather than a terrifying judgment, you're not suppressing fear that has already emerged. You're changing the cognitive interpretation that would generate fear in the first place. Your amygdala then processes exciting challenge instead of terrifying judgment, naturally producing a different emotional response with less need for effortful control. The capacity for emotion regulation evolved relatively recently in evolutionary terms and develops relatively late in individual development. Many animals with functional limbic systems lack the sophisticated prefrontal regulatory capacity that humans possess. This regulatory capacity represents one of the defining features of human emotional life, the ability to feel powerful emotions yet choose whether and how to act on them. Understanding this system reveals that emotions and rationality aren't opposites locked in conflict, but rather integrated systems where higher cortical regions can guide and shape the emotional responses generated by ancient subcortical structures. Yet emotion regulation, sophisticated as it is, doesn't operate in isolation from memory. The same emotional experiences your prefrontal cortex helps regulate are simultaneously being encoded into memory by the hippocampus, with emotional significance marked by amygdala activity. How you regulate emotions in the moment influences not just your immediate experience, but also how those events will be remembered. Strongly regulated emotional experiences may be remembered differently than unregulated ones, with different emotional coloring and different levels of detail preserved. This intricate dance between emotion and memory reveals another layer of complexity in how feelings shape your experience, not just in the present moment, but in the stories your brain tells about your past. Every significant moment of your life exists in memory because emotion marked it as important. The day you fell in love, the moment you heard devastating news, your graduation, the birth of a child, a narrow escape from danger. These experiences remain vivid years or even decades later, while countless ordinary moments fade into forgotten background. This selective preservation isn't random. Your brain actively prioritizes emotionally charged events, ensuring they're encoded more strongly, consolidated more thoroughly, and retrieved more easily than neutral experiences. This emotional enhancement of memory emerges from the intimate partnership between two structures sitting side by side in your temporal lobes: the amygdala, which processes emotional significance, and the hippocampus, which forms new memories. The hippocampus, shaped like a seahorse curving through each temporal lobe, serves as your brain's memory formation center. When you experience something new, sensory information flows into your hippocampus, where specialized neurons encode the details of what happened, where it occurred, and when it took place. This structure doesn't store long-term memories permanently. Instead, it acts as a temporary processing station where new memories are formed before being gradually transferred to cortical regions for permanent storage through a process called consolidation. But the hippocampus doesn't operate alone when forming memories. Right next to it sits the amygdala, constantly evaluating whether incoming information carries emotional significance. When something emotionally arousing happens, whether positive like unexpected success or negative like a frightening encounter, your amygdala activates and sends signals directly to your hippocampus. These signals essentially tell the hippocampus this matters, encode this strongly, make sure this memory persists. Research using direct electrical recordings from electrodes implanted in human brains during neurosurgical procedures has revealed the precise mechanism of this interaction. When people successfully encode emotionally arousing information into memory, researchers observe synchronized high-frequency electrical activity in both the hippocampus and amygdala simultaneously. The two structures show coordinated neural oscillations, their electrical rhythms synchronizing in a pattern that doesn't occur during encoding of neutral information. This coordinated activity appears to strengthen the synaptic connections that form memory traces. Even more remarkably, when researchers applied inhibitory electrical stimulation to disrupt this coordinated activity, they specifically eliminated the memory enhancement normally seen for emotional material. People still remembered neutral information normally, but emotionally charged items lost their privileged status. This demonstrates that the amygdala-hippocampus interaction doesn't just correlate with emotional memory enhancement, it causally creates it. The amygdala influences memory through multiple mechanisms beyond direct communication with the hippocampus. During emotionally arousing experiences, your amygdala triggers the release of stress hormones including cortisol and epinephrine from your adrenal glands. These hormones circulate through your bloodstream and cross into your brain, where they activate receptors in both the amygdala and hippocampus. The amygdala, now activated by stress hormones, send modulatory signals to the hippocampus that enhance memory consolidation processes. This hormonal enhancement explains why memories formed during highly emotional states, both extremely positive and extremely negative, show such exceptional durability. The surge of stress hormones creates a neurochemical environment optimized for memory formation, ensuring that emotionally significant events are stamped into long-term storage with unusual strength. Studies have shown that administering stress hormones after learning enhances memory consolidation, while blocking stress hormone receptors impairs the memory advantage normally seen for emotional material. The evolutionary logic behind emotional memory enhancement becomes clear when you consider survival in ancestral environments. An animal that remembers which situations proved dangerous, which foods caused illness, which actions led to rewards, and which locations provided resources will survive and reproduce more successfully than one that treats all experiences as equally memorable. Emotion serves as a tag marking, "This experience matters," and your memory systems respond by prioritizing those tagged experiences for long-term storage. This emotional prioritization explains the vivid memories people form during traumatic or life-changing events. Psychologists call these flashbulb memories because they preserve exceptionally detailed, vivid snapshots of emotionally intense moments. People often remember exactly where they were, what they were wearing, who they were with, what time of day it was, and numerous other contextual details when they heard about major public events or experienced personal crises. The intense emotional arousal triggered widespread amygdala activation, which in turn ensured that every available detail was encoded strongly by the hippocampus. Yet emotional memory isn't always beneficial. The same mechanism that helps you remember important information can create persistent, intrusive memories of traumatic experiences. When something genuinely threatening or horrifying happens, the massive amygdala activation and stress hormone surge create extremely strong memory traces. These memories can become so robust that they intrude into consciousness unbidden, triggered by subtle reminders of the original trauma. This underlies post-traumatic stress disorder, where a normal emotional memory enhancement mechanism produces memories so strong and easily triggered that they disrupt daily functioning. The amygdala's role in fear conditioning demonstrates emotional memory formation at its most basic. In fear conditioning, a neutral stimulus like a tone is paired repeatedly with something aversive like a mild shock. After just a few pairings, the tone alone begins triggering fear responses. Your heart rate increases, your muscles tense, you experience subjective feelings of anxiety. This learning happens primarily in your amygdala, specifically in the basolateral complex where information about the tone and the shock converge. When these two types of information arrive simultaneously at amygdala neurons, the synapses connecting them undergo long-term potentiation, a strengthening of the connection between neurons that forms the cellular basis of memory. This creates a memory trace encoding the association between the tone and danger. Once formed, this association proves remarkably persistent. Even years later, hearing a similar tone can trigger the learned fear response, demonstrating the durability of emotionally conditioned memories. Research using precise lesions in animals has mapped this fear memory circuit with remarkable detail. Damage to the lateral nucleus of the amygdala prevents animals from forming new fear associations entirely. They still feel pain from shocks and hear tones normally, but they cannot learn that the tone predicts danger. Damage to the central nucleus produces a different effect. Animals form the association normally, but cannot express appropriate fear responses. This dissociation reveals that different amygdala subregions handle learning versus expression of fear memories. The hippocampus contributes contextual information to emotional memories. While the amygdala encodes that something dangerous or rewarding happened, the hippocampus records where and when it happened, who was present, and other environmental details. This contextual encoding allows appropriate generalization. You might fear dogs after being bitten, but specifically fear large dogs in parks more than small dogs in homes if the hippocampus preserved contextual details about where the bite occurred. Damage to the hippocampus impairs contextual fear memory while leaving simple fear conditioning intact. The infant who smiled indiscriminately at all faces now shows stranger anxiety, becoming distressed when unfamiliar people approach, evidence that their amygdala has learned to distinguish known caregivers from unknown individuals and to treat the unknown as potentially threatening. Emotional memory begins forming during the first year as the hippocampus matures enough to encode episodic information. Infants develop preferences based on past emotional experiences, becoming excited at the sight of a favorite toy, showing distress when a doctor appears because previous visits involved uncomfortable vaccinations. These early emotional memories lack the detailed contextual information mature hippocampal function provides, but they demonstrate that the amygdala-hippocampus interaction underlying emotional memory enhancement operates in rudimentary form even in infancy. The second year brings dramatic emotional development as toddlers begin acquiring language and showing self-conscious emotions. Around 18 months, children first recognize themselves in mirrors, demonstrating emerging self-awareness that creates the foundation for emotions like embarrassment, pride, and shame that require understanding oneself as a social object others evaluate. These self-conscious emotions depend on prefrontal cortical regions that process self-referential information, showing early signs of the prefrontal development that will continue for 2 more decades. But emotional regulation remains extremely limited in toddlerhood. The terrible twos earn their reputation because toddlers experience powerful emotions without the prefrontal regulatory capacity to modulate them. A minor frustration triggers a full meltdown because the dorsolateral prefrontal cortex that implements cognitive control strategies remains dramatically underdeveloped. The anterior cingulate cortex that monitors for emotional conflict and helps select appropriate responses shows limited activation in toddlers, leaving them at the mercy of automatic emotional responses generated by mature limbic structures. Parents must provide external emotional regulation, soothing, distracting, removing the child from overwhelming situations because the child's internal regulatory capacity hasn't yet developed. Preschool years bring improving emotional understanding and rudimentary regulation attempts. 3 and 4-year-olds begin recognizing emotions in others, labeling feelings with words, and understanding that people have different emotional responses to the same situations. These advances reflect strengthening connections between emotional processing regions and language areas, and early development of theory of mind capacities implemented by temporoparietal and medial prefrontal regions. The preschooler who covers their eyes to avoid seeing something scary demonstrates primitive emotion regulation, an avoidance strategy that requires recognizing emotional states and attempting to control them, even if the strategy proves ineffective. Middle childhood from ages 6 to 12 brings substantial advances in emotional regulation as prefrontal cortical regions undergo rapid development. The dorsolateral prefrontal cortex begins implementing cognitive control strategies more effectively, allowing children to redirect attention away from emotional stimuli, use self-talk to manage feelings, and apply simple reappraisal strategies like reminding themselves that a medical procedure will help them feel better. Brain imaging studies show increasing prefrontal activation during emotion regulation tasks across middle childhood, correlating with behavioral improvements in children's ability to control emotional expressions and cope with negative emotions. Yet compared to adolescents and adults, school-age children still show relatively limited emotional complexity. They tend to categorize emotional experiences in simpler terms, happy or sad, mad or scared, without the nuanced recognition of mixed emotions that requires more mature prefrontal integration. When asked if they can feel happy and sad at the same time, young children typically say no, insisting emotions are mutually exclusive. This reflects the hierarchical organization of emotional processing you encountered in mixed emotions. Their limbic structures process emotions in relatively binary ways, while prefrontal regions capable of holding contradictory information simultaneously remain immature. Social emotional processing advances considerably during middle childhood as children become increasingly sophisticated at reading others' emotions, understanding different perspectives, and navigating peer relationships. The superior temporal sulcus, fusiform face area, and other social perception regions show adult-like activation patterns by late childhood. But integrating social emotional information with appropriate behavioral responses continues developing as prefrontal limbic connectivity strengthens. The child who recognizes that a classmate feels sad may still struggle to generate an appropriate comforting response, demonstrating the gap between emotional perception and emotionally guided social behavior that closes gradually across development. Adolescence brings perhaps the most dramatic and challenging emotional developmental period. During the teenage years, limbic structures undergo significant reorganization while prefrontal regulatory regions continue maturing at different rates, creating a developmental asynchrony that profoundly affects emotional experience. The amygdala shows heightened reactivity to emotional stimuli during adolescence, particularly social emotional cues like peer evaluation, rejection, and social status. Brain imaging studies reveal that adolescents show stronger amygdala responses to emotional faces than children or adults, suggesting a period of peak emotional reactivity. Simultaneously, the dopamine reward system undergoes major changes during puberty. The nucleus accumbens and ventral tegmental area show heightened sensitivity to rewards, making positive experiences feel more intensely pleasurable, but also increasing vulnerability to risk-taking and reward-seeking behavior. This heightened reward sensitivity explains why adolescents pursue intense experiences, become more susceptible to peer influence, and sometimes engage in dangerous behaviors despite understanding the risks intellectually. Their reward systems drive powerful approach motivation that their still maturing prefrontal regulatory systems struggle to modulate effectively. The prefrontal cortex continues developing throughout adolescence, but different prefrontal regions mature at different rates. Sensorimotor regions mature relatively early, followed by association cortices, with the prefrontal regions implementing cognitive control and emotion regulation among the last to reach full maturity. This protracted prefrontal development means teenagers possess nearly adult-level emotional processing and limbic structures paired with still developing regulatory capacity in prefrontal regions. The result, intense emotions with limited ability to regulate them effectively, creating the emotional intensity and volatility that characterizes adolescence. Structural brain changes during adolescence reveal the mechanisms underlying these functional shifts. Gray matter volume in prefrontal regions actually decreases during the teenage years as synaptic pruning eliminates unused connections, making remaining circuits more efficient. White matter increases as myelin sheaths code axons, speeding neural transmission between distant brain regions. These structural changes reflect the brain optimizing its architecture, eliminating excess connections from childhood while strengthening the most used pathways. The specific pathways that strengthen depend heavily on experience, meaning the emotional brain adolescents develop reflects not just biological maturation, but also their particular emotional experiences and regulation attempts during these formative years. The developmental gap between emotional reactivity and regulatory capacity creates vulnerability during adolescence. Mental health problems including depression, anxiety disorders, eating disorders, and substance abuse often emerge during the teenage years, partly reflecting this imbalance between mature emotional processing systems and immature regulatory systems. Adolescents experience emotions as intensely as adults, but lack fully developed tools for managing overwhelming feelings, creating risk for maladaptive coping strategies. Understanding this developmental vulnerability helps explain why adolescence represents a critical period for learning effective emotion regulation strategies that can protect mental health across the lifespan. Social emotional processing becomes paramount during adolescence as peer relationships assume central importance. The social brain networks involving the temporoparietal junction, superior temporal sulcus, and medial prefrontal cortex show heightened activation when adolescents process social information, particularly social evaluation. Teenagers become acutely sensitive to peer opinions, social status, and potential rejection, reflecting both hormonal influences on social motivation and developmental changes in neural circuits processing social rewards and threats. This intense social focus served developmental purposes. Adolescence represents the period when humans historically began forming relationships outside their family of origin, requiring sophisticated social-emotional skills to navigate peer groups and establish new social bonds. By the early 20s, prefrontal regulatory systems approach mature functioning, though subtle refinements continue into the late 20s. The dorsolateral prefrontal cortex implementing cognitive control, the ventrolateral prefrontal cortex selecting appropriate regulatory strategies, and the medial prefrontal regions monitoring for emotional conflict all reach adult levels of connectivity and efficiency. This prefrontal maturation brings improved emotion regulation, more sophisticated understanding of mixed emotions, better integration of emotional and rational decision-making, and enhanced capacity for delaying gratification. The young adult possesses both fully mature emotional processing systems and fully developed regulatory capacity, creating potential for optimal emotional functioning. Yet emotional development doesn't stop at adulthood. Across the adult lifespan, emotional processing continues changing, though these changes reflect experience and neuroplasticity rather than dramatic structural maturation. Research on emotional aging reveals that older adults often show improved emotional regulation compared to younger adults, despite some decline in prefrontal cortical volume. This positivity effect describes how older adults preferentially attend to and remember positive emotional information more than negative, showing better emotion regulation and greater emotional well-being even as cognitive abilities decline. Brain imaging studies suggest this improvement reflects strategic shifts in how older adults deploy attention and process emotional information. Rather than relying primarily on effortful prefrontal regulation strategies, older adults may develop more efficient regulatory approaches based on decades of emotional experience. They've learned which situations trigger unwanted emotions and can avoid or modify those situations proactively rather than requiring effortful regulation after emotions arise. This experiential wisdom accumulated over a lifetime creates emotional benefits that compensate for or even exceed losses from age-related neural decline. The amygdala shows reduced reactivity to negative emotional stimuli in older adults compared to younger adults, while maintaining responsiveness to positive stimuli. This shift in amygdala processing contributes to the positivity effect, reflecting either structural changes in the amygdala itself or altered input from prefrontal regions that have learned to filter emotional information differently. Based on life experience, the specific mechanisms remain debated, but the phenomenon demonstrates that emotional brain function continues adapting throughout life rather than remaining static after development completes. Individual differences in emotional development reflect both genetic factors and environmental experiences. Twin studies reveal that emotional reactivity, regulation capacity, and vulnerability to mood disorders all show significant heritability, demonstrating genetic contributions to emotional brain development. But environmental factors profoundly shape how genetic potential unfolds. Childhood adversity, trauma, secure versus insecure attachment relationships, and chronic stress all influence how emotional circuits develop, creating lasting differences in amygdala reactivity, prefrontal limbic connectivity, and stress hormone regulation. This developmental plasticity creates both vulnerability and opportunity. Adverse experiences during sensitive periods can establish patterns of heightened emotional reactivity and impaired regulation that persist into adulthood. But positive experiences and interventions can also reshape emotional processing, strengthening regulatory circuits and reducing maladaptive reactivity. Psychotherapy, particularly approaches teaching emotion regulation skills, can produce measurable changes in prefrontal limbic connectivity even in adulthood. This demonstrates that while genetic factors and early experience shape emotional brain development, neuroplasticity allows continued change across the lifespan. Understanding emotional development reveals that the brain processing your feelings today emerged from a decades-long journey of neural maturation, synaptic refinement, and experience-dependent plasticity. The amygdala that detects threats, the hippocampus that binds emotions to memories, the prefrontal cortex that regulates feelings, all these structures developed gradually, shaped by both biological programs and your particular emotional experiences. This developmental perspective transforms how you understand emotions themselves, revealing them not as fixed responses, but as products of systems that continue adapting throughout life. Every emotional experience you have changes your emotional brain slightly, strengthening some synapses and weakening others, reinforcing certain patterns and creating new possibilities. The fear that once paralyzed you can diminish through exposure and new learning. The joy that once eluded you can emerge as reward circuits respond to changed circumstances. The regulation that once felt impossible can become more automatic as prefrontal circuits strengthen through practice. Your emotional brain isn't fixed but living, growing, changing with each year of experience. This realization brings us to the final chapter of this journey, integrating everything you discovered about the emotional brain into a coherent understanding of what it means to be a feeling human being, living with these ancient systems and modern regulatory capacities, experiencing emotions not as obstacles but as essential guides shaped by millions of years of evolution and your own unique developmental journey. You've traveled deep into the architecture of emotion, exploring structures that emerged with the first mammals over 200 million years ago. and cortical systems that developed far more recently in evolutionary time. You discovered how the amygdala detects significance in milliseconds, how neurotransmitters paint your emotional world with color and intensity, how the hippocampus binds feelings to memories, how the prefrontal cortex allows you to consciously shape emotional experiences that would otherwise shape you. Now, as this journey reaches its quiet conclusion, these separate insights can merge into a single understanding: what it means to be a human being who feels. Every emotion you experience emerges from this intricate system: ancient limbic structures operating automatically beneath awareness, neurotransmitter cascades creating physiological states, cortical networks transforming raw reactions into conscious feelings you can name and understand. When anxiety tightens your chest, that's your amygdala detecting potential threat, and your hypothalamus triggering stress responses evolved to prepare your ancestors for danger. When joy spreads warmth through your body, that's dopamine flooding reward pathways that reinforced beneficial behaviors for millions of years. When grief weighs on you after loss, that's your emotional memory systems recognizing that something profoundly significant has changed, demanding acknowledgement and adjustment. These feelings aren't random noise interfering with rational thought. They're information, sophisticated signals carrying wisdom accumulated across evolutionary time and your personal lifetime. Your amygdala has learned through countless experiences what situations require vigilance. Your hippocampus has encoded which people and places brought joy or pain, creating emotional memories that guide current choices. Your ventromedial prefrontal cortex integrates this experiential knowledge into somatic markers, bodily feelings that steer decisions before conscious analysis completes. Dismissing these signals as irrational means ignoring information your rational mind cannot access directly. Yet emotions aren't infallible guides. They can reflect outdated learning from situations that no longer apply, triggering fear of dangers that have passed or attraction to rewards that now bring harm. The same amygdala that protected your ancestors from predators can generate anxiety about social situations that pose no genuine threat. The same dopamine system that motivated pursuit of survival-critical resources can drive addictive behaviors. The same emotional memory enhancement that helps you learn from significant experiences can create intrusive traumatic memories that disrupt daily functioning. Emotions evolved to serve survival in ancestral environments, not necessarily to create happiness in modern life. This is where your uniquely human cortical systems transform emotional experience. The prefrontal regions that developed most recently in evolution allow you to step back from automatic emotional reactions, to recognize when feelings may not match reality, to consciously choose whether to act on emotions or regulate them. You can feel anxiety yet proceed toward important goals. You can recognize anger rising yet choose a measured response rather than lashing out. You can notice sadness without letting it eclipse joy still available in other aspects of life. This metacognitive capacity, the ability to observe and think about your own emotional states, creates a distinctive human form of emotional freedom. Understanding the emotional brain reveals that feelings and reason aren't enemies but partners. Your emotions provide rapid assessments of significance, compress experiential learning into immediate signals, connect you to others through empathy, and motivate behavior toward what matters. Your rational cortical systems offer perspective, evaluate whether emotional responses fit current circumstances, imagine alternative futures, and implement regulatory strategies when automatic reactions prove unhelpful. Optimal functioning harnesses both, feeling emotions fully while thinking about them clearly. The richness of human emotional life emerges from this integration. You can experience mixed emotions, bittersweet nostalgia, anxious excitement, proud grief, because cortical regions hold contradictory feelings simultaneously rather than forcing binary choices. You can regulate overwhelming emotions through reappraisal, changing how you interpret situations to alter their emotional significance before reactions fully develop. You can connect deeply with others through empathy, your insula and anterior cingulate cortex simulating their feelings and creating genuine emotional resonance. You can make complex decisions by integrating emotional wisdom with rational analysis. These sophisticated emotional capacities distinguish human consciousness. Your emotional brain continues changing throughout life. The systems processing your feelings today differ from those you possessed in childhood and will differ from those you'll have in older age. Every emotional experience subtly rewrites your neural circuits, strengthening some pathways while weakening others. The fears you overcome through exposure lose their grip as amygdala-based associations weaken. The joys you cultivate through attention strengthen as reward circuits respond to practice. The regulation strategies you develop become more automatic as prefrontal circuits refine through use. You're not stuck with the emotional brain you inherited. You're actively shaping it with every choice about how to respond to feelings. This developmental perspective offers hope. If emotional difficulties arose partly from how your brain developed in response to early experiences, then new experiences can continue reshaping these systems. The heightened amygdala reactivity associated with anxiety can decrease through mindfulness practices that strengthen prefrontal regulatory circuits. The reduced reward sensitivity contributing to depression can improve through behavioral activation that re-engages dopamine pathways. The emotional dysregulation that disrupts relationships can improve through therapy, teaching regulation skills that create new neural patterns. Change remains possible because your brain retains plasticity, the capacity to reorganize in response to experience, across the entire lifespan. Living skillfully with emotions means recognizing them as neither masters to obey blindly nor obstacles to overcome completely. They're evolved systems providing valuable information that deserves attention while also sometimes requiring conscious modulation. When anxiety surfaces before an important event, you can acknowledge the signal without accepting its catastrophic interpretation. The feeling might reflect appropriate activation preparing you for challenge rather than accurate prediction of disaster. When anger rises in response to perceived injustice, you can honor the signal that something violates your values while choosing whether aggressive action serves your goals. When sadness follows loss, you can allow the grief its natural course rather than demanding immediate return to positive emotions. Different situations call for different emotional strategies. Sometimes feeling emotions fully without attempting regulation proves most adaptive. Grief needs to be felt. Joy deserves full experiencing. Love benefits from unreserved expression. Other times, conscious regulation serves you better. Anxiety spiraling into panic benefits from reappraisal. Anger threatening valued relationships calls for modulation. Fear based on outdated associations needs updating through new learning. Emotional wisdom involves knowing which approach fits which circumstance, and that knowledge develops through experience. The social dimension of emotion adds another layer of meaning. Your feelings don't exist in isolation, but arise partly from and influence your connections with others. The mirror mechanisms that create empathy mean other people's emotions activate your own emotional circuits, creating genuine resonance rather than merely intellectual understanding. The attachment systems that bond you to loved ones use the same dopamine reward pathways that respond to food, making social connection feel fundamentally rewarding. The social emotions like shame, pride, guilt, and gratitude depend on understanding how others perceive you and comparing those perceptions to your standards. Emotions weave the fabric of social life, coordinating cooperation, signaling intentions, and creating the felt quality of relationships. Understanding this social emotional architecture reveals why relationships profoundly affect well-being. Supportive connections activate reward systems and buffer stress responses, literally changing your brain's emotional functioning. Chronic conflict or isolation activate threat systems and impair regulation, creating vulnerability to emotional difficulties. The people you spend time with shape your emotional experiences not just through what they say and do, but through the neural systems that automatically resonate with their feelings. Choosing relationships wisely means recognizing their power to influence your emotional brain. The emotional wisdom that often develops with age suggests what skilled emotional living might look like. Older adults frequently show improved emotion regulation despite some neural decline, suggesting they've learned efficient strategies through decades of experience. They attend preferentially to positive information, avoiding unnecessary engagement with negative stimuli, demonstrating proactive regulation rather than reactive control. They've developed intuitions about which situations trigger unwanted emotions, and can structure their lives to minimize such triggers. This accumulated experiential knowledge creates emotional benefits that compensate for age-related biological changes. You don't need to wait for old age to develop this wisdom. Every time you pause before reacting emotionally to consider whether your feeling matches the situation, you're exercising and strengthening prefrontal regulatory circuits. Every time you acknowledge difficult emotions without being overwhelmed by them, you're expanding your window of tolerance. Every time you notice emotional patterns and adjust your environment or interpretation accordingly, you're developing the proactive regulation that characterizes emotional maturity. The emotional brain you'll have in future years depends partly on how you work with emotions today. Living with feeling means accepting that you're fundamentally an emotional creature who also possesses rational capabilities, not a rational being occasionally disrupted by emotion. The limbic structures processing your feelings emerged over 200 million years ago and operate continuously, mostly outside awareness, evaluating significance and preparing responses. The cortical systems that create conscious awareness of emotions and allow their regulation developed far more recently and require more effort to engage. Your default mode is emotional. Feelings arise automatically from ancient circuitry. Conscious emotional regulation represents an achievement, a recent evolutionary capacity that requires deliberate activation. This recognition invites self-compassion. When emotions feel overwhelming, when regulation fails, when you react in ways you later regret, these aren't personal failings but reflections of how your brain works. You're operating with systems designed for survival in ancestral environments, overlaid with regulatory capacities still maturing well into your 20s and requiring conscious effort to implement. Perfect emotional control represents an unrealistic standard. Skillful emotional living means working with the brain you have rather than demanding it function differently. The neurotransmitters flowing through specific pathways create the felt texture of emotional experience. Dopamine's reward signals, serotonin's aversive processing, norepinephrine's arousal. These chemical messengers blend in varying proportions like primary colors mixing to create the full emotional spectrum. Understanding this neurochemical foundation demystifies feelings. When depression creates anhedonia, that's depleted dopamine reducing reward sensitivity. When anxiety generates physical tension, that's norepinephrine activating stress responses. These aren't character flaws but neurochemical states that can change. The emotional memories stored through amygdala-hippocampus interactions shape current experience more than most people realize. Past situations resembling present circumstances trigger associated emotions automatically, often before conscious recognition of the similarity. This can create wisdom, learned caution around genuinely risky situations, attraction to authentically rewarding experiences, or maladaptive patterns when past learning doesn't apply to current reality. Noticing these automatic emotional responses and asking whether they fit present circumstances engages the metacognitive capacity that allows updating. The decisions you make draw heavily on emotional information, whether or not you acknowledge it. Somatic markers guide choices by creating bodily feelings about different options, steering you toward what past experience suggests will work and away from what past experience suggests will fail. Sometimes these emotional signals prove wiser than conscious analysis, detecting patterns your rational mind cannot articulate. Other times they reflect learning that no longer serves you. Learning to distinguish helpful emotional guidance from outdated programming represents a key emotional skill. The capacity to feel what others feel through empathy creates both connection and vulnerability. Your anterior cingulate cortex and insula simulate others' emotional states, generating genuine vicarious experience rather than merely conceptual understanding. This shared neural representation makes other people's suffering genuinely painful and their joy genuinely uplifting. It binds you to others through feelings more than thoughts, creating the emotional foundation of compassion and cooperation. But it also means others' distress affects you directly, requiring boundaries to prevent emotional overwhelm. As you settle towards sleep tonight, you can rest in this understanding. Emotions aren't chaos to control, but information to integrate. The tightness in your chest signals what matters. The warmth in your heart reveals connection. The ache of loss honors what was meaningful. The flutter of excitement points toward possibility. These feelings flow from systems sculpted across millions of years of evolution and decades of your personal development, operating with wisdom deeper than conscious thought alone can access. Your emotional brain works constantly, beneath awareness, guiding you through life with signals refined across evolutionary time. The amygdala watches vigilantly. The hippocampus binds experience to memory. Neurotransmitters paint feelings with chemical precision. The prefrontal cortex offers perspective when you need it. All these systems working together create the rich emotional landscape of human experience. This is the end of your journey. May you rest in the understanding that what you feel is part of being beautifully, completely human.